Hi students, in this class let us quickly revise the biological classification chapter. Every year in NEET examination we could expect 1 to 2 questions from this chapter. To start with, classification of living organisms was born out of need of living organisms for different purposes by human beings like food, shelter and clothing. He classified living organisms just as organisms used for food, organisms used for shelter, and organisms used for clothing, but this is not a scientific method. The first scientific method of classification was started by Aristotle. He used simple morphological features for classification of living organisms. Based on that, he classified plants into 3 groups: trees, herbs, and shrubs. And he classified animals into majorly 2 groups which contain red blood are called anima, and animals which do not have red blood, he called them as anima. Later, another scientist, Carolus Linnaeus, you could remember from first chapter as well, living world, he proposed a binomial system of nomenclature, and also he has a contribution in proposing 2 kingdom classification. He used criteria gross morphological features for classification of living organisms, and he classified all living organisms into 2 kingdoms, that is kingdom Plantae and kingdom Animalia. Kingdom Plantae include all plants and kingdom Animalia include all animals. And this system of classification, that is 2 kingdom system of classification, was used for a long time. But this has some drawbacks. Let us see what are the drawbacks of Linnaeus system of classification. It did not distinguish between eukaryotic organisms and prokaryotic organisms, as well unicellular and multicellular organisms, and it did not distinguish between photosynthetic algae and non-photosynthetic fungi into 2 different organisms. They are considered as one and they are classified in kingdom Plantae, and large number of organisms did not fall into either of the group. So the classification system was considered, it has to consider other characters other than the gross morphological classification by other scientists. So they proposed we have to use other characters like cell structure, nature of cell wall, mode of nutrition, habitat, method of reproduction, and even evolutionary relationships or phylogenetic relationship between the organisms, along with the morphological features which were used by Linnaeus. So later on, using all these, classification system was evolving, or it has undergone several changes over the period of time, and it is not been constant in any period of the time. But in all the systems of classification, plant and animal kingdom are being the constant one. Later on, after proposition of Linnaean system of classification for a change, R.H. Whittaker in 1969, he proposed 5 kingdom classification, and he classified all living organisms into 5 kingdoms like kingdom Monera, Protista, kingdom Fungi, Plantae and Animalia. And he used 5 criteria. They include cell structure, body organization. Remember these criteria considered by R.H. Whittaker are very important. You have to remember cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationship between the organisms are the main criteria used by R.H. Whittaker. So remember these 4. Later, Carl Woese in 1990 proposed 3-domain classification. Here he divided kingdom Monera into 2 domains like archaea and bacteria, and other eukaryotic kingdoms, that is kingdom Protista, fungi, Plantae and Animalia, were classified under eukaryota or eukarya. So hence archaea, bacteria and eukarya are the 3 domains. As I said earlier, classification systems were not being the same over the period of time. It is been changing. For that, there are some issues and considerations that have influenced the classification. For example, in earlier systems of classification, bacteria, blue-green algae, fungi, mosses, fern, gymnosperms and angiosperms, they all of them are found to be having cell wall. Hence, they are altogether placed in one kingdom, that is kingdom Plantae. But here you have to see one issue. Bacteria is prokaryote, blue-green algae is eukaryote. Chlamydomonas is unicellular, Spirogyra is multicellular. So here there is a morphological and as well structural difference between the organisms, and also fungi is heterotrophic, green plants are autotrophic, but all of them are having cell wall, and even between fungi and green plants there is a difference in the cell wall composition as well. Fungal cell wall is made up of chitin, plant cell wall is made up of cellulose. So when such considerations are made, which I mentioned earlier, fungi to be placed in a separate kingdom, that is kingdom fungi. All prokaryotes are placed in one kingdom, that is kingdom Monera. Unicellular eukaryotic organisms are placed under Protista. So here Chlamydomonas, Chlorella. Chlamydomonas and Chlorella are unicellular eukaryotic organisms. Amoeba and Paramecium are unicellular eukaryotic organisms that lack cell wall. So earlier they were placed in animal kingdom, but upon some changes or considerations, they were placed in kingdom Protista. So now let us start with 5 kingdoms proposed by R.H. Whitaker. To start with kingdom Monera. Kingdom Monera include bacteria as the sole members, and bacteria are the most abundant microorganisms found on earth, and they occur everywhere. The habitat of bacteria, they occur everywhere. And based on morphological structure of bacteria, bacteria are classified into 4 major groups. I hope all of you are familiar with those four groups, and you have to remember the names with specificity. Spherical bacteria are called coccus or cocci, and rod-shaped bacteria are called bacillus or bacilli. And rod-shaped bacteria usually contain spores within them, which are the resistant structure produced in harsh environmental condition. Comma-shaped bacteria are called Vibrium or Vibrio. Spiral-shaped bacterium are called Spirilla or Spirillum. Here, Spirillum have flagella, which are the locomotory structures. Bacteria, they are very simple in structure, as we learnt earlier. They may be having 4 different basic shapes, but they are very complex in their behavior because of extensive metabolism taking place in their body. Remember, this question could be asked for assertion and reason type of question. So here assertion will be bacteria are simple in structure, complex in behavior, and reason would be because of extensive metabolic reactions taking place in them. Now let us look at quickly classification of bacteria. Bacteria are mainly classified into 2 types, archaebacteria and eubacteria. Archaebacteria are considered as special bacteria because they live in harsh habitat like extreme salty areas, and such groups of archaebacteria are called halophiles. They may live in hot springs and they are called thermoacidophiles. They may live in marshy areas, they are called methanogens. And even methanogens could be found in cattle's stomach, that is rumen of cattle, and they are helpful in production of biogas. And in second year microbes in human welfare chapter, you have studied methanobacterium are helpful in digestion of cellulose. So archaebacterial group can survive in harsh environmental condition because of their different cell wall structure. Now let us move on to eubacteria. Eubacteria are called true bacteria because they have rigid cell wall and flagellum if they are motile. Eubacteria are classified into 2 groups, autotrophs and heterotrophs. Autotrophs, as we say, they are capable of synthesizing their own food, but synthesis of food will be different. Based on that, they are classified into 2 groups. First one is photosynthetic autotrophs. As the name suggests, they perform photosynthesis. Example for photosynthetic autotrophs are cyanobacteria or blue-green algae. They contain chlorophyll A, and this pigment will be similar to higher plants. Cyanobacteria are unicellular or colonial forms or filamental form. And habitat, they may present in freshwater or marine water or terrestrial environment. And colonial forms of photosynthetic autotrophs form gelatinous sheath on their body. and they even form algal blooms in polluted water bodies. So here especially water bodies which are present near to the residential areas have green color because of pollutant present in them. Using that pollutants or organic matter, cyanobacteria, colonial forms will start to grow and make the entire body to appear in green color. So example for cyanobacteria here is nostoc and anabina. They can fix nitrogen in a special cells called heterocyst. So you have to remember a diagram of nostoc which is given in NCERT textbook, including labelings. Gelatinous sheath is there, heterocyst is there. Then second group of autotrophs are chemosynthetic autotrophs. Here they are called chemosynthetic autotrophs because they oxidize various types of organic matters like nitrates and ammonia and use the released energy to synthesize ATP. Remember, oxidation of the organic matter directly will not produce ATP. Energy will be utilized for synthesis of ATP, and they play very important role in nutrient cycling of nitrogen, phosphorus, iron and sulfur. And the second group of bacteria is heterotrophic bacteria. Heterotrophic bacteria are most abundant bacteria. Remember, most abundant organisms are bacteria. Most abundant bacteria is heterotrophic bacteria. And they are very important decomposers. They play very important role in human affairs. That is, they are being the part and parcel of human life. There are positive effects as well negative effects. So let us talk about positive effect. They are helpful in making curd from milk. The organism used will be lactic acid bacteria. They are used for production of antibiotics, and even the negative effect is they are the pathogens of human crops and farm animals as well pests. To quote some diseases caused by bacteria: cholera caused by Vibrio cholerae, tetanus caused by Clostridium tetani in human beings, and citrus canker is used in caused in plants by bacteria Xanthomonas citri. Then coming to the last part of this topic, that is bacteria reproduction. Generally, reproduction in bacteria takes place by fission, where the bacterial cell will divide into two as such to form two cells. And spore formation will take place during unfavorable condition, as we discussed earlier in rod-shaped bacteria. There is a sort of DNA transfer taking place that is called conjugation. There is formation of conjugation tube between two bacterial cells through sex pili. The replicated DNA will transfer to the other bacterium. The another group of bacteria placed in Monera is mycoplasma. Mycoplasma, they don't have cell wall, so they are the smallest living cell which are not having cell wall. With the cell wall will be bacteria, without cell wall will be mycoplasma, and they can survive without oxygen, and they cause diseases in plants and animals. So in next video, let us continue with kingdom Protista. Thank you.